Okay, so we are in the digestive system right now, and this video is going to be about carbohydrates. So carbohydrate digestion starts in the mouth. We know that. And it starts with salivary amylase. That starts the chemical digestion of carbohydrate. There is no digestion going on of carbohydrate in the stomach, so it resumes in the small intestine. Now, carbohydrates are big molecules, so they have to be broken down into smaller polysaccharides, and then down to disaccharides, and then ultimately to monosaccharides to actually be absorbed in the body. So the salivary amylase breaks that down into the smaller polysaccharides. And then once it's in the small intestine, pancreatic amylases break that down into the disaccharides. And once we have disaccharides, and that's maltose, lactose, and sucrose, then we have brush border enzymes, or enzymes that are in the brush border, that break those down to monosaccharides. So that would be maltase, lactase, and sucrase. So once we get down to glucose, fructose, and galactose, those are what get absorbed into the body. Okay, so glucose and galactose are absorbed into the body through secondary active transport. Active transport. So it's kind of a process to get those two molecules in. Fructose is a little bit different, and it enters the cells passively, so through passive transport. Okay, so if I draw this out, I can show you how this basically occurs. So I'm going to draw my cells here, and this area up here is the lumen. So that's where the food is. This is the tube of the small intestine. So remember, that's where we are right now. The disaccharides, the maltose, the lactose, and the sucrose, are changed into monosaccharides along what we call the brush border. And that's where these enzymes are in the brush border. And those enzymes change or break down those disaccharides into glucose and galactose and fructose. So I'm doing them different colors because they go into the cell differently. So the glucose and galactose move in actively. So they use secondary active transport. And you're going to see a lot of these mechanisms that we use are going to utilize sodium because there's lots of sodium in the outside of the cell, we know. So for glucose and galactose, there is a transporter on the membrane that's called SGLT, S-G-L-T. And that means sodium glucose transporter, okay? And those are on the membrane, and what they do is they utilize the gradient for sodium to drag in the glucose and galactose. So we've got sodiums here, and they are coming in, and with that comes the glucose and the galactose. Okay, so as sodium is coming in the cell, we don't want it to build up, because if it builds up, then it'll stop flowing. Okay, so we have to do something to keep the gradient alive for that sodium. So to do that, we have to utilize something called sodium potassium ATPase, or the sodium potassium pump. And that sodium potassium pump will take the sodium out and we'll put it in the blood. And I need to put a little blood supply here. Same color, and I'm sorry, but here's my little blood. So it pulls that out into the blood and brings in potassium. And that will maintain this gradient to allow sodium to flow in readily, and it will bring in the glucose and the galactose through secondary active transport. The fructose is different. So the fructose comes in passively. So there is a channel, sort of like a leaky channel, that allows for the movement of fructose to come into the cell. Okay, there's always more fructose on the outside than on the inside, so it's going to come through diffusion. Okay, so once inside the cell does not consume the glucose. The galactose is converted to glucose. Some of the fructose is converted to glucose, and once that is done, there's another transporter in the basolateral side, which is called GLUT. So that's specific for glucose. And there are different types of GLUT transporters. You don't need to know which ones are what, just to let you know that. So you'll see those often. So those come through and go into the bloodstream. Okay, so from there, of course, the glucose goes into the blood and it travels to the liver where it is processed and then dispersed throughout the body. Okay, now this, if you were wondering, probably weren't, but this potassium is coming into the cell and is getting built up. So what happens to that? Well, you should know, hopefully, that there are lots of leaky channels for potassium in the cellular membrane of all cells. So the potassium just continues to diffuse out as it normally would. So it's coming in through the sodium potassium pump, and of course it is going back out, and that's what would be normal anyway for it. This cell just, I did this lumpy up here. I want you to understand, and I probably should have gone ahead and put what this was, but this is microvilli. And that's what makes up this brush border. So the brush border there has enzymes in it that, of course, are going to break down the disaccharides into the monosaccharides, and that's how we absorb those. And this is how we absorb those monosaccharides that are left there. Now it's interesting the glucose comes in, it isn't consumed by the cell. The cell does not, these cells do not eat glucose or require glucose for energy. They eat something called glutamine. So that keeps them from utilizing this glucose, and all of the glucose that comes in there goes into the blood and onto the liver. So that is how carbohydrate absorption occurs. The next video we'll talk about protein absorption.